Only this last little bit to go, just mitosis. Hopefully this is a review for most of you. Okay, life cycle of the cell, we're talking about interphase and mitosis. Cells are always in one or the other, interphase or dividing. In interphase, you actually have two states you can possibly be in. It can be in normal metabolic activities. Most of the life of the cell is going to be spent being a cell, being a part of the tissue, being operational, contributing to the good of the organ. But sometimes it'll get a cell signal and it'll say it's time to prepare to divide. So that is also interphase. Preparation for cell division is its own state of interphase. Then when interphase completes, we get our mitotic phase. That is the actual act of cell division. And of course, these phases, mitotic phase has subphases, interphase has subphases, which we need to talk about to a degree of detail. So interphase, G1, S, and G2. What is not illustrated is G0, which is our process of just being a cell, our normal cell activities. Then we get the signal to divide and we enter G1. So G1, I hear it sometimes as gap phase, gap phase 1, or growth phase 1. I prefer to think of it as growth phase 1 just because it's more descriptive, but gap is technically the technical term. So in growth phase 1, the cell literally starts to grow. It starts to increase its organelles. There's also, not illustrated here, a checkpoint. The cell is going to check on its DNA, and if the DNA is mutated, then the cell may undergo apoptosis, which is programmed cell death. If something is too wrong with the cell will kill itself for the betterment of the tissue. If you know what the definition of out-of-control mitosis is? Out-of-control mitosis is cancer. So we actually have several mechanisms in place to prevent mutated cells from becoming cancer. It's only when those mechanisms go awry that we actually do develop cancer. So that's just something to think about for future lectures for pathophysiology, for pharmacology, aging course, things that are coming down on the line. But for now, all you need to know is about G1 is growth phase 1. S phase is our synthesis phase. In this phase, we double our DNA. We replicate our DNA. A bunch of enzymes go in and they copy the DNA blueprint onto 2 new copies of the DNA, doubling the amount of DNA that you have. In this phase, the DNA has to be loose. This is why I was telling you about chromatin earlier on in the lecture. Chromatin is the loose form of DNA in which it can be copied, in which it can be synthesized. That occurs during S phase. G2 is growth phase 2. We double our organelles. We finalize our preparation for mitosis. By the end of G2, we should have twice everything. We should have double the organelles. We should have double the DNA from all of these processes in interphase. So here you can see a cell that is an interphase. You can tell because of the appearance of the nucleus here and the chromatin. You can see that the DNA inside of it is just loose, as opposed to condensed into chromosomes. In a minute, we'll show you what a cell looks like with chromosomes as opposed to chromatin. Remember, those are just 2 different forms of DNA. So the mitosis phase, if everything checks out, there aren't too many mutations in the genetic code, everything looks good, we've doubled our DNA, then we can begin the mitotic phase. 2 distinct events occur in this phase, mitosis and cytokinesis. The mitosis is the one that's cut down into prophase, metaphase, anaphase, and telophase. So in order to remember this, it's kind of goofy, it's kind of gross, it's kind of weird, but sometimes those memory tools that are gross and weird are the ones that actually stick, the ones that actually work. If you can visualize something weird to help you remember it, you're more likely to remember it when it actually counts. So I remember pee mat. I remember a guy peeing on a mat. Once you visualize a guy peeing on a mat, super gross, you're not going to forget it. You can't possibly forget the order that these things happen in. Prophase, metaphase, anaphase, telophase. So at that point, you've got all of your vocab in order. All you have to do is remember the basics of what happens in each phase. And again, this is clinically relevant. There are medicines that act on mitosis. For example, cancer medications, colchicine, are going to act at right around metaphase to prevent further mitosis. It's pretty cool. Our understanding of the cell, again, gives way to our understanding of medicine. So prophase, metaphase, anaphase, telophase visually looks like this. Cytokinesis is our final division of the cytoplasm, splitting of the plasma membrane. We have formed two daughter cells that should be genetically identical to the progenitor cell back here. In prophase, the chromatin, the loose DNA, coils and coils and coils and coils in on itself to form chromosomes. If you look very closely right here, you may want to look at this in your PowerPoint and zoom in on it to get some detail. Instead of loose stringy DNA, you see these condensed chromosomes. You see these sort of solid blocks. They're kind of stained a little bit deeper purple here. That's how you know we're entering into mitosis and we are out of interphase. So prophase is going to be our first phase. In it, we still have, by and large, one cell, but the nuclear membrane is beginning to break down. The chromatin has condensed into chromosomes. Our chromosomes are taking this interesting form of sister chromatids. They're held together in a region at the center called the centromere. I know everything starts with C, so be very careful with your mnemonic devices, your memory tools for memorizing this. Line up all of the C words and differentiate them for yourself is what I recommend. You see these spiky things here in the cell, those are our spindle fibers. This is a modified type of cytoskeleton. The spindle is going to guide mitosis. It's simply forming in prophase. And the end of prophase is when the nuclear envelope is completely dissolved. You should not see a clear nuclear envelope at the end of prophase. Metaphase is my personal favorite just because histologically, cytologically, it's so easy to recognize because you have this thing called the metaphase plate. And the metaphase plate, this is actually chromosomes lined up at a nice, neat equator. And you can see the mitotic spindles. There's one here and there's one here. So even though you still have one cell, you can see the direction that they are actually going to be pulled apart into. So the spindle fibers, that cytoskeleton that's modified to guide mitosis, is attaching to that centromere that's holding together the sister chromatids. So again, the sister chromatids being your two copies of DNA that you got from S phase. In anaphase, you'll notice that the metaphase plate has split. You've got one sister chromatid going this way, you've got one sister chromatid going that way. Each daughter cell should get one copy of the same piece of DNA each. That's what makes it genetically identical. At this point, the cell is elongated, but is not actually splitting just yet. That happens in telophase. You get something called the cleavage furrow in telophase. Yes, cleavage, haha, and that's exactly why. So cleavage being the place where things are cleaved in two, but complete division does not happen in mitosis, it happens in cytokinesis. But you can see the organelles get pulled to either side. The cleavage furrow appears, it will complete cytoplasmic division. Again, cytoplasmic division being division of the cytoplasm and the stuff in it, the organelles. The chromosomes may begin to uncoil in this phase. So we're going back from chromosomes to chromatin. And towards the end of this, the nuclear envelope is going to begin to reform around our genetic material. Finally, cytokinesis completely divides the two, and it goes back into interphase. Each new daughter cell begins interphase all over again. Hopefully it's going to spend some time being a functional cell within a tissue, contributing to the health of the organ and the activities of the organ. And that's really it. That's our mitosis. That's all we need to know about mitosis for this course. Aging in the cell, I mentioned apoptosis is programmed cell death. If the cell in G1 or G2 detects something awry with the DNA, it may commit apoptosis or programmed cell death. Necrosis is different because it's from an outside agent, a toxin, a mechanical damage, temperature, something like that happened to the cell and it became necrotic and it died. This is more harmful to the tissue. Apoptosis does not cause inflammation. Necrosis does cause inflammation. So again, aging is a normal and continuous process indicated by changes in number of organelles or chromatin structure. There's a lot more to it than that. You will see plenty more of that in your future A&P and pathophysiology courses. We don't need to cover that in detail here. All right, if we were in class today, we would have lab time coming up. For week 1, lab time is not extensive. It's time to practice your regional anatomy. It's time to practice your positional terms, your directional terms, and any leftover time we would have spent going back over worksheets. We would have spent more time coloring. We would have tried to make it as fun as possible, and time for questions and answers and things like that. So I do recommend doing all of those things. Oh yes, and modeling cells out of clay. I do recommend that you do all of these lab activities at home. Try to keep up with this, try to keep on top of the content. If you leave these lectures for right before the exam, it's probably not going to go as smoothly. So I hope you all have enjoyed your snow day. I hope you have plenty of time this afternoon to internalize these lectures. If you have any questions, feel free to email me, reeves.binford@plattcolorado.edu. Thank you so much for your time this afternoon.